All right, team. Today we begin a new unit. One of my favorite things to lecture and teach upon is the kingdom fungi, and that's where we're headed. We can scroll down and see we are in module 4, and we will pop into this fungi PowerPoint and begin our discussion. One of the fascinating things about the fungi really is that we don't quite know what to do with them. They're kind of oddballs in many ways. If we string some words together, and I'll probably even add a few more to begin our discussion of this group of organisms that is really nothing like plants. And why are we studying the fungi in botany? Traditionally, the fungi have been studied by botanists. The specific field is called mycology. Myco in Latin means fungus, but botanists in general have always studied the fungi. What we're going to learn in here is that they're really, if they're related to anything, they're more likely related to animals, not plants. As my mycology teacher in grad school told me, he said, you know, when there was the primordial ooze of life and all the things were evolving, all these fingers were poking up out of the ooze, the fungi likely had their own finger, and it was a thumb. That's what he would say. Okay. So, you know, what differentiates this group of organisms from other kingdoms? They are eukaryotic. They are exclusively heterotrophic. No one has ever found a photosynthetic fungus, although they are sensitive to light in many ways and require light in certain ways to grow normally. They are filamentous in terms of their life form, with the exception of the yeasts, which are unicellular. And we're going to add a few more words to this. We're going to say they are, if we want to be really strict about our definition, that they are non-motile. They don't have flagella. And then we're going to learn that there's this organism called an oomycete, a water mold, and it does have flagella. But if we want to be strict about our definition of what a fungus is, we're going to say non-motile. The other thing that makes them all in the same group is that they all produce spores. So they're spore producing. Some other general things about them: cell walls of chitin. And this molecule chitin is very interesting as it appears in the cell walls of fungi. The fungal filament is a little bit strange as well, because if these are nuclei, what we see in many of the fungi when we look at them is that there are no septa. They're aseptate. In other words, there's not a cross wall separating one nucleus from the next, which is just very strange. I mean, how does this nucleus know what territory it controls? I don't know how to answer that. Okay. But this cell wall material is chitin. And why this is interesting is because now, if we consider this, we have a very clear biomolecular link between the fungi and not plants. But where in the world do we find chitin? Chitin forms the exoskeleton of arthropods. What are arthropods? Arthropods are insects, spiders, crustaceans like shrimps and lobsters and crabs. The shell of a lobster is the same biomolecule as the cell wall of a fungus. So that is a clear biomolecular link that links the fungi, not to plants, but to animals. And I often tease my vegan friends when they're about to scarf down that big portobello sandwich. I'm like, You're eating your ancestors. They don't want to hear that. They're half starved. Okay. It's Sunday night, y'all. Sunday night. We're doing some kingdom fungi. Okay. So that's a good little intro for you, I think. Now, as far as the taxonomy goes, I'm just going to go ahead and tell you this. You know, it gets pretty complicated. I mean, if you look at the what splitters want to do, they want to split this thing up into so many different groups. I don't see, for our purposes, the practicality of that. This is an older taxonomic scheme which uses 4 divisions, or you, given the information I just told you, might want to call them phyla. I don't care. You can call them either one, phylum or division. It's the same level. But considering these things are probably more animal-like than plant-like, it might make sense to call them phyla. Okay. This is an older taxonomic scheme. It's a simplistic one. If you care to learn the most latest taxonomic scheme, I'm sure you could look it up, okay? But for the purpose of teaching this material, it's just a convenient way to break them up. You have all of them ending in mycota. Again, that means fungus in Latin. So we have the zygo, asco, basidio, and deuteromycota, okay? And you can see what's in each of these groups. The zygomycota are the molds and mildews, everyone's favorite. No. The ascomycota include the cup fungi and the yeasts. The basidiomycota include the mushrooms, toadstools, shelf fungi, stinkworms, puffballs. There should be a comma after that. Sorry about that. Let's do that. Let's put a little comma here, because a puffball and earth star are 2 different things, okay? And then finally, we have the last group, the deuteromycota, which we call the fungi imperfecti, or the imperfect fungi. The basis of this taxonomy, sexual reproductive structures. And so each of these first 3 groups makes a very specific structure or structures when they do sex, when they do fusion and meiosis to get to their next generation. The last group doesn't have sex, or no one has ever observed it. No one has ever taken the time, or else they just have lost the ability to reproduce sexually. They're very good at asexual reproduction. They make lots and lots of spores through mitosis. And so we'll talk about this, the imperfect fungi, and how they're kind of a garbage group in the sense that because we don't know what structure they make or would have made, we don't know where else to put them, okay? But they could very well include members of any of these 3 groups. They're most often allied with the ascomycota. And so some people want to get rid of the deuteros and put them all in the ascomycota based upon very similar asexual reproductive strategies. But for our purposes, we're going to separate them out. Okay. And then at the end, we'll talk about things that are on the fringe, like slime molds and water molds. They're sort of fringe organisms. Okay. They don't quite fit. All right, next slide. Now we get to the zygomycota. And oh, you are so familiar with these. You may not know it, but you are so familiar with these, with this particular organism that is a zygomycete. The most common zygomycota is ubiquitous. It's found worldwide. It's found in every imaginable habitat and place. And this sucker is called Rhizopus stolonifer. And there are other species, but Rhizopus stolonifer is black bread mold. And we call it black bread mold because it loves bread, but it also loves your nectarines, as you can see here. What you're looking at, remember, they're filamentous organisms. The white is the filaments, right? and the black is the spores. Anytime you see a black and white fungus, it's likely going to be rhizopus. Okay. How is it relevant? Well, gosh, how is it not relevant? Agent of spoilage, right? How relevant is that? Well, how many loaves of bread have you thrown away? How many rotten strawberries? How many times all the people on the earth that have done the same thing? And you begin to realize, wow, we're talking about, I don't know, billions of dollars worth of wasted food. What happens when rhizopus gets in the grain silo and, takes it out? So as an agent of spoilage, rhizopus is very much relevant to us. Okay. How else is it relevant? It is toxic. Do not eat rhizopus. Don't eat moldy bread. You can't cut the mold off of bread, generally speaking, especially black and white mold. What you see is the mold is probably grown into the porous bread. Unlike cheese, where you can wipe it off or cut it off, it's a little more difficult with bread. Don't feed moldy food to animals. It's toxic to them too, including songbirds. Don't put moldy bread out for the birds. Throw it away or compost it. Okay. Allergens. Rhizopus spores are known allergens. When you go and have an allergy test where they do the scratch test, one of usually 2 or 3 fungi that they test you for will be rhizopus. Many people are allergic to the spores. Here's a big one that you might not think about. Contaminants. I didn't spell that very well. Let's do a little better. Contaminants of sterile culture. All over the world, there are sterile labs where they're growing bacteria or other microbes, maybe other fungi for research or science. What happens when one of these little spores, boom, gets into the petri plate? Well, what happens is it contaminates the petri plate, and then you have to throw that petri plate away. You lose that culture. How did the spore get there? Well, it's interesting. Same thing you could think about your moldy bread. How does the mold start to grow on the bread? The bread is hot and it's put in a bag, and you would expect it to be a relatively sterile environment. You are usually the source of contamination. The spores are probably stuck on your grubby little fingers, and when you reach into that loaf of bread to pull out your bread for your PB&J, you inoculate the loaf, right? How long does it take for the mold to start growing once you inoculate the loaf? Well, it depends on the bread. If it has preservatives in it, it may never grow, or it may take a really long time. But if you're talking about bread from Earth Fare, it's going to mold in a few days, start to mold in a few days in the summer. Okay. So contamination of sterile culture, in these sterile labs, if there's a contamination issue, they call in the audit police. Literally, the contamination police come and they try to find the source of the contamination, and it better not be you. If you work in one of these labs, you literally have to be clean. I mean, you scrub your fingernails and shower and keep yourself clean so that you're not a contaminant of the lab. Okay. Here is a close-up of rhizopus. I actually took this picture. We're going to do a life cycle for rhizopus in just a bit. But what you're looking at here is a piece of bread. I just moistened it and left it out. It grew a nice colony of rhizopus, as you can see. And again, all these little black dots are not individual spores, but what we call sporangia. And I'll go ahead and introduce that word to you. And a sporangium has a little stalk like this, and then it has a little head, and it's full of spores, lots and lots of spores. So the spores are in the head there. And so this isn't magnified enough to see individual spores. What you're seeing are individual sporangia. Okay. So that's kind of cool. We'll do a life cycle here in just a second. Okay. Okay. Next slide. This is another zygomycete that is definitely worth talking about. It's called Pilobolus, and it's also commonly called shotgun fungus, and I'll tell you why we call it that, and then we can actually even watch a little video. It is in the Zygomycota. It is a dung fungus. Common name, shotgun fungus, genus is Pilobolus. I'll just go ahead and tell you, you're not really responsible for these genus names. I give you a genus as an example, within a kingdom and division, in this case, a division or phylum. The level that we're studying this stuff on is actually, I think that's actually a Pilobolus. Yeah. The level that we're studying this on is really on the division level. And so I give you this just for convenience in case you want to look it up. So here's the thing. These live in dung. They live in cow poop. And if you know anything about cows, you know that a cow basically won't eat right around its patty. When it poops, it knows better because it knows there's probably parasites in the poop. And so generally the cow will stray away from a cow patty when it's eating, when it's feeding on its grass. Well, this organism, the shotgun fungus, needs to get its capsule of spores into a cow. The spores don't hurt the cow. This isn't a parasite of cows. It's a saprophyte. It lives in dung. And so what this is full of spores, a sporangia here. And so what Pilobolus does is a strategy. We have a pressure building up in this little tube here, hydrostatic pressure. And it's also heliotrophic, so it follows the sun. And so you can see how it's sort of angling toward the sun. And what it's going to do, the pressure will build up so great that it shoots, it launches this capsule full of spores away from the patty where it's fruiting, where it's making its spores, so that the cow will ingest it and thus continue the cycle. So that's a pretty wacky strategy. And they've done all this research. They found that the velocity of these things when they're shooting is higher than the velocity of a handgun in some cases. So we can let's just for fun have a little aside. Let's hop out of here and let's watch a video of Pilobolus just so it is just so it's a little more streamlined here. Here, let me see here. Stand by. Let me clear it. And let me out. Let me have a new tab. Let me go to YouTube. Pilobolus. And I'm sure somebody has videotaped this thing shooting off its capsules, and that's what we'll. Fungus cannon in super slow motion. That one's 5 minutes long. Whew, I don't know if we can watch for 5 minutes, but this one is actually very cool. I think it's set to, I think this one is set to classical music, which is kind of cool. Okay, there you have it. Now you can say you've seen the shotgun fungus shooting its sporangia cannon. Okay, so what makes it in the zygomycota? That's the next thing we have to discuss and think about, and also that leads us into looking at the life cycle of this organism. Okay. Well, the structure that it makes is a structure that's formed during sexual reproduction, and that structure is called a zygospore, sometimes called a zygosporangia. I don't know what the deal is with that. Some books call it a zygospore and some call it a zygosporangia, but you can bet it's this dark thing in here. That's the zygospore. That says zygospore, I promise. There we go. Zygospore or zygosporangia. Either one, you can call this. This is the structure that's made during sexual reproduction. We're about to do a life cycle. Okay, let's do the life cycle. What the heck? Let's do her. Okay, we're going to do the asexual and the sexual life cycle. I suppose we could do it on this slide, but it's a little awkward. Let's go to a whiteboard type scenario. I think that makes more sense. Wonder what that is. Okay, so how does this thing reproduce? How does it make its, next generation? Asexual is very successful through sporangia formation. And what does that look like? Well, the filaments grow out in all directions. They digest food. And then eventually they start making sporangia with spores. And what's the process here? Mitosis times many. Okay, so, this thing just divides kind of uncontrollably as it digests its food and grows out in all directions. And then when it starts to, reach its sort of maximum potential, you might think of it that way, it doesn't take very long. It's just a few days at the most. It could be even less than that before it starts making sporangia. Each of these little spores can then be released. They become airborne and they start to grow into new filaments. Super successful strategy, right? You could have a loaf of bread and it could have 5 or 6 generations of sporangia, asexually produced sporangia that all came from one spore. Well, what do you know about it? If it's mitosis, then genetically they're all theoretically identical aside from any sort of mutation that might have occurred, right? So it's just cloning itself, essentially. It's very, very good strategy. Okay. What about sexual? Well, these organisms aren't male and female. Some things to understand about the fungi. First thing to understand about them in terms of their sexual life cycle is they're haploid dominant. Okay. And what does that mean? Well, it means that the vegetative phase of the life cycle is haploid. It's 1N. Okay. So think about the implications of that. There are mating strains, and they're pretty simple in something like Rhizopus. It's just a plus and a minus, kind of like what we saw in the Protista. So I'm going to do them different colors, but I'll just go ahead and tell you if this is, let's say, a filament, and this is supposed to be a spore here, and it's germinating. Okay. If this is a plus, I can do a minus in a different color. Go Tigers. And, I'm drawing cross walls. I probably, oh, shoot. Probably shouldn't be drawing cross walls in all reality. Oh man, let's just start over, y'all. Come on. Let's just start over. Here we go. Here's a spore. The filament is grown out. These are nuclei. We'll let this be a plus strain. But yeah, I don't like Clemson. Okay. Oh, Tar Heels, baby. It's my daughter's alma mater. Okay. And we'll let this be a minus strain. You can't tell them apart. I'm drawing them different colors. Only Rhizopus knows if it's a plus and a minus or not. Okay. What happens though over time? Dang, nation. I think it's not working good for me. What happens over time is when two appropriate strains come physically in contact with one another, a wall will form like this. Okay, so now we have these two strains and they just about touched one another like this. And when they sense the presence of each other, they'll form a wall there. At that point, guess what? These are going to become gametes. Remember, this thing is already haploid, 1M. Okay. Now, in the fungi, the gametes are literally just nuclei. Okay. That's all a gamete needs to be. It's already haploid. Fusion occurs, and that's where we get the zygospore. So the zygospore is really just a zygote, and we just call it a zygospore. So the spore part implies that it's a resistant. I'm going to put some of this color in there too, because it's really a mixture of these two, right? Okay. The term zygospore implies a resistant stage. And in fact, what usually happens, as long as there's plenty of food, Rhizopus is more than happy just to reproduce asexually. You know what I'm saying? And just make spores because it's very successful. It's really when the environment starts to go bad on Rhizopus that it is triggered into this sexual reproductive thing. And that makes sense. Again, we talked about this before. If the environment sucks, why not recombine genes and come up with a better genotype for that given environment? Okay. So now, how do we get back to haploid? This is the zygotic life cycle. And so the next thing that happens is meiosis. Now, the life cycle can be arrested in the zygospore stage, and the rest of all the mycelium, all the fungal filaments can die. They can die off. And then just we're left with these zygospores, which then eventually the life cycle can crank back up when the environment gets better, and this thing will start to, it'll continue on. It'll do meiosis. When it does meiosis, we get 4 cells, right? Turns out that we get both mating strains inside here, okay? And then a curious thing happens. From these 4 cells, outgrow sporangia out of the zygospore. And there'll be more than one usually. And there'll be spores in here. And there'll be spores of both mating strains. What that tells us is at least 2 of these cells must be doing the mitosis that it takes after meiosis to make these spores, right? If only one of these 4 cells was doing it, we'd only get one of the mating strains, but we get both mating strains. So that means at least 2 of them are involved, and it could be that all 4 are involved. Unfortunately, we don't really know the answer to that because we can't see inside the zygospore. It's very, it's very opaque. You can't really see inside of it, okay? And then, of course, these spores are released. We'll draw both colors, a little Chapel Hill in here. Okay, and then whoop, they germinate and you're back where you started. This is the zygotic life cycle, right? We've seen it before. Only diploid cell is the zygote, which we're calling a zygospore. Just be clear, it's the zygote, same thing. It's just zygospore is saying it's a resistant zygote, okay? Well, I realized there are two words that I didn't tell you that are very important when you're dealing with fungi. One of the words is hyphae. Let me do this in a different color. Hyphae are fungal filaments. So that's a hyphae and that's another hyphae. The individual filaments of a fungus are called hyphae. Now, in mass, when you have a bunch of hyphae, they can all stick together and form these sort of rope-like structures. And so masses of hyphae, we have another word for, and you've probably heard this word, mycelium. Mycelium is when you have masses of these hyphae all stuck and matted together to form, they can form three-dimensional structures like a mushroom, for example. Okay, so both of these words, very important, hyphae and mycelium, are two words that I should have introduced at the beginning when we were on the first slide, but that's okay, now you know. Okay, and now you know how Rhizopus does sexual reproduction. What makes it, what makes a zygomycota in the zygomycota? Because it makes this thing called a zygospore when it has sex. Okay. All right. Next group is the ascomycota. And this is a really fascinating group, y'all. I'm so excited to tell you about this group. It includes some of the most important edibles on Earth, certainly the most valuable edibles on Earth in terms of edible fungi. The morels we have here, and they're safe to say our most valuable, pound for pound, our most valuable edible mushroom, if you're talking about monetary. And then the truffle, which we don't really have too much of around here. There are some out west, but primarily truffles are European. Italian and French and Spanish truffles are extremely valuable. And we'll talk about the fact that, you know, truffles can fetch $500 an ounce for dried. Okay. This group includes the toxic ergot fungus and all of the ethnobotanical significance of ergot that we're going to talk about. You're going to be fascinated by it. Okay. This group also includes many plant pathogens that cause what we think of as blight. And, you know, anytime you see this term blight, when it's referring to a disease of plants, it's typically a fungus, or we would like to, you know, use it to describe a fungus, fungal blight. And so, for example, the Irish potato famine, if you're sitting in this room and you're of Irish descent, you may be sitting in this room because of an ascomycota, you know, because your ancestors were starving and they're like, we're getting off this rock, we're going to the New World. Okay. Also Dutch elm disease, American chestnut blight, which is the chestnut blight was considered the single greatest ecological disaster of all time in the United States, if you can believe that, or at least in the Southeast. And then most recently, we have a dogwood anthracnose, which is killing our native dogwood trees. Not killing them quite as fast as they thought, but killing them nonetheless. So that's just a little taste of what you can expect to learn in this part of the discussion on fungi, the ascomycota. I'm not sure why I didn't capitalize that. I should have. Make it a big A. There we go. Okay, now I'm going to get rid of it. Next slide. The morel. So we commonly call members of the ascomycota the cup fungi. And in the case of the morel, there are a bunch of little cups on what looks like a cap of a mushroom. Usually, you know, if someone looked at this, they would call it a mushroom. You know, mushrooms we like to reserve for the Basidiomycota, but It's fine. You get the idea. So, these are edible. This is a yellow morel, Morchella esculenta, which is esculenta means delicious in Latin. Okay. And these will pop up out of the ground for a very short time. Oops, shoot. Touched my pen and look what happens. Okay, well, now we're all over the place. Let's go back. Okay. I can just do the annotate and then it won't change the slide. That's helpful. So, when's the truffle or the morel season? April. You could just think of the month of April as going to be probably when you're going to find them, if you're going to find them. And under mixed hardwoods, especially ash. If you want to find yellow morels, go look under ash trees. Morels are fetching about $75 a quarter pound last year. That's $300 a pound. That's pretty good take. There are people that wander around in the woods and find these things and, they make some money. Aside from that, you can find them yourself and eat them. They're delicious. They're one of the safer ones that you can collect. They're relatively easy to identify. I will point out, do a little research. There is something called a false morel, which is toxic. And so you want to make sure that you identify them. I always tell people, there's a lot of people crazy about wild mushrooms. And I tell people, because, I'm constantly, you could imagine people send me a picture. Is it this? Is it this? I no longer ID them for people by a picture. And what I do tell people is even if that's what it is, get someone else that knows fungi to also say that that's what it is. And then you're probably going to be okay. So morels are generally considered to be one of the safer wild mushrooms that you could collect. And we'll see a picture of a false morel in a little while. Okay. And these are truffles. So the truffle has a long history of collection and use by humans. Particularly in Europe, there's a truffle season, and traditionally, what have they used to find truffles? Dang nabbit. That's so annoying. Traditionally, what animal have they used to find truffles? They have used the pig, right? Particularly the male pig. And you could imagine leading pigs through the woods. Smell truffles. It was quickly learned that pigs, for some reason, had an affinity to these mushrooms or these fruiting bodies. The thing about the truffle is that what you're looking at here, these are full of spores. They're called sclerotia, and they're fruiting bodies that are full of spores, except in the case of truffles, normally you would expect the spore-producing structure to break through the earth and be above the ground. But in the case of the truffle, they're buried in the ground like this, okay? And so you can't see them. It's hard to find them. And so what people discovered was that for some reason, pigs were very attracted to these truffles, attracted to the smell of them. And a good truffle pig could sniff out truffles from a mile away. And so the idea is you lead the pig through the woods, it sniffs out the truffles, it starts digging and scratching on the ground, and then you pull the pig back and you dig out these truffles. Well, scientists were really curious why these pigs were so interested in these small fungi that are buried in the ground, certainly not for their nutritional value. And they began to do a little research about the smell of the truffle. And what they found was that the smell acted as a pheromone to the pig. A pheromone is a chemical attractant. Now, you're familiar with pheromones if you ever have had a cat or a dog, a female cat or dog that wasn't fixed and they went into heat, and they produce a smell that attracts all the male animals to the location, right? Well, when they got to analyzing this pheromone-like chemical that these mushrooms were producing, they realized it was almost identical to the pheromone that a female pig, a sow, produces when she's in heat. So these pigs don't think they're going after mushrooms. They think they're going to get them some, you know, and it must be a little unnerving when they get to the spot where the smell's coming from. And the pig's like, she's buried in the ground, you know, and the pig starts to try to dig the mushroom up. Okay. Or tries to dig the pig up, but it's really a mushroom. And then as a consolation prize, the pigs will dig these up and chew on them and gnaw on them, okay? So what's going on here? Well, it's probably an incredible example of convergent evolution for these fungi to produce this chemical. You could imagine the dilemma for the fungus is its spore producing body's buried in the ground. It needs to get those spores airborne. What better way to get it airborne than to attract a large animal to dig you up? And so over evolutionary time, these chemicals probably randomly evolved and they became attractive to the pig. And the more similar to the smell of the pig, the more likely it was to get dug up and the more likely it was to transmit its spores and continue on that line, okay? Well, pigs are big and hard to deal with. And so, you know, what's a better choice? How about a dog? And yes, now they use dogs. And so they have truffle dogs, they train them. There's actually, believe it or not, a guy in right outside of Durham that trains truffle dogs. Most of them get sent to Europe. And, you know, the dog is a little easier to deal with. You give the dog a treat and that's all the dog expects, okay? What about the truffle in terms of the way it tastes? Some people despise them and other people love them. They have a garlicky, very strong garlicky, sort of body odor, stinky feet smell, which, you know, I mean, garlic is stinky, but it is appealing as a culinary device. So you may have had truffle oil. They will infuse oils with truffles. As far as like eating a whole truffle, you know, they're really expensive. The most expensive truffle ever was a guy spent 75,000 euros for one truffle, and it was about this big. And this was this man that obviously had more money than he knew what to do with, and he was just being, you know, being indulgent. But generally speaking, you know, they go for about $500 a dried ounce, not a pound, but an ounce. So they're quite valuable. Okay. No one has ever quite figured out how to grow them on a large scale. There are people working on it. There's a guy in Charlotte that's working on it to grow them on hazelnuts. And he's had some success, and now he sells inoculated hazelnut bushes that he says you plant them and, you know, 7 to 10 years later, you can start harvesting your truffles off of them, which would, you know, break the market open, so to speak. This was the same guy that figured out how to grow morels and made millions of dollars growing morels. So he does have a background in the field. Okay. My one experience with actual truffles was when I was in France and I was at a fancy French restaurant and I ordered some kind of creamy soup a la truffle. And I was I was kind of expecting the truffles to be like dumplings in the soup, but instead it was just grated little shavings on the top. But it was quite delicious. So that's the story of the truffle. And I think we're going to stop there, but be sure to tune in next time because we're going to talk about the ergot fungus after we talk about cup fungi. Okay. All right, team. I hope you've enjoyed the lecture. This is going to be fun, and I'll talk to y'all real soon. Okay. I hope you have a great day, and yeah, take care.